Use of microalgae as a sustainable alternative to reduce contamination levels in the Atoyac River. Based on the research we made, we decided to focus on reducing the levels of cadmium in the Atoyac River by expressing the GSH2 gene in a microalgae strain. This gene is found in E. coli and encodes for glutathione synthetase for the accumulation of cadmium using the PLBr19 plasmid.
The Atoyac River has a pH between 6.6 to 8, however in rainy seasons a pH generated between 7.6 and 8.4, in dry seasons its pH is 7.8 to 8.7 respectively. On the other hand, the average annual temperature in the region is 70.22 degrees Celsius. In this way, the temperature of the Atoyac River in the rainy seasons is between 90 to 22 degrees Celsius, while in the dry seasons it is between 90.8 to 23 degrees Celsius. Taking into account the physical parameters of the Atoyac River, it was determined to work with the microalgae Cruella vulgaris. During photosynthesis, it multiplies exponentially its carbon dioxide, sunlight, water, and some minerals. Cruella can grow in various types of culture media and has a great adaptability to different environmental systems. Cruella vulgaris grows in media with a low level of dissolved oxygen and a different pH range. However, previous studies have reported that the pH value in which this microalgae grows best is between 6.3 and 9, while the growing temperature is 23 and 26.5 degrees Celsius respectively.
Our methodology includes 12 steps for the extraction of the gene, construction of the vector, microalgae transformation, and its verification. First, the RNA must be extracted from E. coli. Since we are only interested in the extraction of the coding sequence for the GSH2 gene, RNA will be extracted instead of DNA. The gene must be inserted in a PLBr19 plasmid that includes the CAMB35S promoter and the CAMB terminator to allow the introduction of coding sequences between them. The construction of this vector was based on the previous research done by Strom. This vector has a neomycin phosphotransferase gene that causes acanamycin resistance. The P70 promoter and T2 terminator stimulate the production of the GSH2 gene, and the TNOS terminator and PNOS promoter stimulate the production of the neomycin gene. In this construction, the nopalin terminator and promoter are presented in inverted positions to express the antisense nopalin synthetase RNA, which can inhibit the expression of mRNA to reduce resistance to antibiotics and reduce their consumption. However, since we are not interested in this process, our vector won't include the TNOS and PNOS sequences presented in the image. To create the desired vector, a process of reverse transcription PCR has to be performed to first convert the RNA into cDNA and then copy our gene of interest. The decision to use a two-step RT-PCR was made because it saves reagents, it is more sensitive, and it can provide templates for multiple PCRs. Now, some specific primers are needed to copy only the gene of interest. These primers were designed to anneal to both sides of the GSH2 gene. A mix of the target DNA, dNTP, stacked DNA polymerase, and the primers must be inserted into the thermocycler, and many cycles will be produced until enough DNA has been created. The proper methodology has to be followed to cut the plasmid and gene with the two restriction enzymes. Then, agarose gel electrophoresis is used to separate the DNA fragments by size. UV light is used to identify the desired gene fragment, and gel purification is used to isolate it. Next, ligation must occur to ligate the insert to the plasmid. A reaction must be prepared, microcentrifuged, incubated, and chilled. Now, the vector should be ready to be introduced into the bacteria. This is a diagram of the expected resultant plasmid. The exact modified PLBr19 sequence couldn't be found, which is why the diagram is based on the PUC19 plasmid. Since the PLBr19 plasmid is a modified version of the PUC19 plasmid, the diagram is a very close representation of the actual final vector, which only has the addition of the CAMB promoter and terminator.
For this project, the A2-methation strain GB3101 will be used because it is a commonly used strain for plant genetic transformation, and it has shown the highest transformation rate in previous studies. For the A2-methation's cultivation, the MGL medium is recommended. In this stage, two types of medium will be prepared. The first one will be a regular NGL medium, and it will be used for the cultivation of regular A2-methations, while the second one will contain the two antibiotics required for plasmid maintenance or selection, which in this case they are kanamycin and ampicillin. This medium will be used for the cultivation of the A2-methations containing the binary vector.
For the introduction of the binary vector into the A2-methaciense strain, we will use the electroporation method, which is based on two processes. First is the preparation of the electrocopetan agrobacterium cells, where the tube containing the MGL medium must be inoculated with a colony of our agrobacterium strain and incubated. This first culture will be used to carry out a second inoculation in the same medium. This will be incubated at the same temperature range but will shake it. The cells will be chilled with ice and then centrifugate. Later, they will be washed with sterilized water to centrifugate. They will be washed again, but now with iced 10% glycerol and spinning again. Finally, a liquid and refrigerant in individual tubes. In second place is the transformation of our agrobacterium cells by electroporation, where in the first place the configuration of the electroporation apparatus has to be adjusted. We will add 5 microliters of our plasmid to an liquid of cells. We will transfer the mixture to a cooled cuvette and place in the support. We will simultaneously press the two pulse buttons. Immediately add 300 microliters of the culture medium to the cuvette. The contents of the cuvette were transferred to the glass culture tube and incubate with shaking for 4 hours. Finally, we will select those transforming cells for antibiotics.
For the preparation of bacterial culture media, nutrients were used. The culture were prepared under a phytoplants and under fluorescent lighting equipment during the light and dark phases. For the preparation of the vitamin solution, it was dissolved separate to the mixed stock solution and stored in amber glass slides. When the stock solution with vitamins was prepared, the pH was adjusted and the solution was then autoclaved. Then, second solution were added separate to the bacterial culture media. The inoculation process consists of adding the strain to the nutrient solution. Each vitamin solution was added. Subsequently, each of the culture were homogenized to measure temperature and pH. The culture conditions of 15 days were at 28 plus and minus 1-2 degrees Celsius with an approximate pH value between 6 and 7.
For the transformation of our microalgae, a single colony of Chlorella vulgaris must be inoculated in TAP liquid medium and incubated in the light for 2 days. After this time, this must be concentrated by centrifugation, so that later the sediment is resuspended in the induction medium containing the agrobacterium culture and incubated for infection for 30 minutes. Following infection, they must be centrifugated again. The resulting pellet will be placed on solidified medium with a bacterial agar and incubated in the dark for 3 days. After time, this co-culture will be washed with a cefotaximine solution followed by centrifugation. The resulting pellet should finally be sprayed on the medium containing kanamycin and ampicillin. These plates will be incubated in the dark for 2 days. After this time, they will be exposed to the light for the selection of the colonies.
Another PCR will be performed to confirm the genome integration of the transferred genes into our chlorella. First, we extract and isolate the genomic DNA of the transformed chlorella with the use of CTAB and beta-mercaptoethanol to 2%. Then, we perform a regular PCR by using the following primers. This should help us determine if the gene of interest was correctly transferred. As a last step, we will detect and quantify the production of glutathione synthetase in the transformed chlorella. In order to do this, an enzyme-linked immunosorbate assay, ELISA, should be performed, using rabbit anti-glutathione synthetase as antibody for the assay.